Hello, my name is Devin Darley, and I'm coming here today to teach you a little bit about the mRNA vaccines. Well, with all the recent talk of the mRNA COVID-19 vaccines, you might be wondering what mRNA actually is and how mRNA vaccines actually work. You've probably all heard of DNA, which is the molecule found in the nucleus of every single cell that stores all of a person's genetic information in the form of genes. But how about mRNA? Now, mRNA stands for simply for messenger ribonucleic acid, and it's just one of many types of RNA found in all living organisms. mRNA is synthesized in the nucleus from a DNA template. Once it's mature, the mRNA strand exits the nucleus of the cell through nuclear pores, and it will travel into the cytoplasm. In the cytoplasm, mRNA is translated into a protein. Proteins are simply biomacromolecules needed by the cell to play many critical roles throughout the body. mRNA vaccines work at their simplest level by directly inserting, you guessed it, an artificial mRNA strand into the body. Firstly, the actual mRNA strand must be produced. In order to synthesize clinical-grade mRNA vaccines in the lab, scientists use a process known as in vitro transcription to make the clinical-grade mRNA. For the vaccine to work, once the IVT, in vitro transcription, mRNA strands are made, they need to be packaged before they can be delivered via inoculation into a person's body. mRNA, on its own, struggles to enter cells due to electrostatic repulsions from the cellular membrane. Now keep in mind that naturally mRNA almost never leaves the cell. Furthermore, IVT mRNA is vulnerable to degradation by ribonucleases, or RNases for short, in the extracellular space, because again, mRNA shouldn't be outside the cell. Due to these negative factors, the IVT mRNA is packaged in lipid nanoparticles, which protects the vulnerable mRNA. The surface of these lipid nanoparticles that encompass the mRNA are often coated in a chemical known as polyethylene glycol, or PEG for short. PEGylation, as this process is known, often improves the vaccine's efficacy by making the nanoparticles much more stable. With IVT mRNA safely encapsulated in pegylated lipid nanoparticles, they are injected into a person's muscle. Some of these muscle cells take up some of the encapsulated mRNA via endocytosis, which means that the cell engulfs it. The IVT mRNA is then released from its encapsulation, and it's translated into the SARS-CoV-2 spike protein. The spike protein then sits on the surface of the cell. The immune system will then detect the spike protein, and it will make antibodies to fight what it believes is a foreign virus, which will prime the immune system to fight the wild virus if and when it encounters it. Well, many of the COVID-19 vaccines currently on the market are mRNA vaccines, such as the Pfizer and Moderna vaccines. They are a relatively new technology. mRNA vaccines, in fact, can hardly be described as a vaccine at all. They'd be more accurately classified as a sort of gene therapy, as unlike traditional vaccines, they themselves do not contain the antigen, but rather they hijack the body's machinery to produce the antigen. Only recently, for COVID-19, was the definition of vaccine changed to include this new generation of mRNA vaccine. Now, I hope that you enjoyed this presentation and perhaps even gained a deeper understanding of the science behind this new class of vaccination. Thank you.